Hey everyone, Raptor Chatter here, and we're going to be going back to this kind of format for the months in review, starting with August 2025. Let's get started. Megabalena saporensis is an early balenid whale, meaning it's a right whale, which has a 9 million year gap in their fossil record. This one fits in pretty nicely in that 9 million year gap, and it fits nicely also in that its size is kind of at the transition of the very small earliest ones, and as they started to increase in size as we get towards the modern right whales. Blastulospongia boalensis is a new microfossil, meaning it's kind of a weird single-celled plankton, but also seems like it's related to sponges. This paper actually provides a new 3D look at all kinds of things, including radiolarans and many of these early fossils, and it helps to explain kind of their early evolution as we get from things that are single-celled and close to animals, but then eventually evolved into animals that are things like sponges and, of course, the more derived ones. Fossil animals were sometimes eaten by insects before being buried, and that means you can have decalcified bone, meaning you're essentially taking the bone and removing the calcium. What this means is in some of those areas we were able to find chemical molt casts, basically the exoskeletons that were shed, of some of these insects that got buried with the bones. These molts then got covered in different minerals that precipitated and rotted away, leaving what we have now, which seems like mostly fairly modern groups that are scavengers even today. There's a pattern in the Caribbean for people to land on an island and everything, or maybe not everything, but most of the large animals die. This island, though, is Sombrero Island in the Caribbean, and there's a fossil site there that has preserved 5 lizards, a snake, a tortoise, a frog, and 2 birds. Meanwhile, this island today is so small that it only has 4 migratory birds that pass through occasionally, and 3 lizard species that live there. This is really interesting because people don't really live on this island because it is just so small, but it does show how these small islands are really important steps in the ocean for animals to reach the larger islands. It wasn't essentially just one main event that pushed all of these tortoises, for example, out to all of these other islands in the Caribbean. They may have done it stepwise, landing on small island after small island. There's also a new flat bug from Burmese amber. Check my video here for more about the ethics around Burmese amber, but this fossil at least shows that about 99 million years ago, the flat bugs were probably already facilitating pollination. And that's because some of these fossils, in fact most of these fossils, have pollen grains associated with them and even attached to their body. Now, does that mean they were collecting nectar? No, they may have been going there for some other reason. But whatever did happen, the pollen got onto their bodies, and if they visited another flower, it would produce pollination, which could have been the early steps for evolving proper pollination by insects. Anapachydiskis haggardi is an ammonite from Canada, and this one is really fortunate for us because we have a full growth series. So you can look at the start and see how the shell started to develop and got larger, but then also how the sutures on the chamber became increasingly more complex as the animal aged. These sutures in ammonites are much more complex during the Cretaceous than the Jurassic or Triassic, and we don't exactly know why they were there, but it is something that is under active investigation, and maybe it is something that allowed them to better exchange gases as they reached larger sizes. There's also a big paper that was a review on mammal brains, and also the brains of the relatives of the mammals, so kind of stem mammals. There's a little bit of everything in here, so non-mammalian synapsids headbutting? Yep. Brain changes with body temperature? Yep. There's just a ton. I could stay on this for a long time, but I am not very great with brains, so I will leave that to the experts to discuss a little more, and you can find this paper down below in the links. Gerpegezhus dania oriundus is a new fish, and it's a pipefish, which is fun because that means it has a goofy, kind of long face. This is from the early Eocene, and it's seemingly a very good representative of the group as they first started to diversify, and shows how some of the northern European seas were connected to a southern European sea through the European archipelago. Now this group seems minor, it's just a fish, but this actually has many very well-known types. Not just the pipefish that we know that I already discussed, but also things like the seahorse belong to this group. So we're getting a better understanding of how this group first started to evolve. Lobtelson feldmani is a new shrimpish-like animal coming from New Mexico. It's technically a Belotelsenidae, but the thing is the fossil record of crustaceans shows that many different animals converged on a similar shrimp or lobster-like body plan, and now the Belotelsenidae are actually extinct. So now just our modern shrimp seemingly are taking over a very similar lifestyle. What's really important though is that their telsons, the tail part, is really important for ID, and they show those two really well in this figure, so we can really understand just how diverse they actually were. And that's important because crustaceans fossilize pretty poorly, and as long as we can start getting a few that have really important details preserved, we can start to understand just what they were doing before there was this big faunal turnover from the Belotelsenids to modern day shrimp. Jianfengia is a fossil arthropod, or at least a close relative of the arthropods from China, and it has wonderful soft tissue preserved, including the soft tissue of the brain. There's also links with the great appendages, think of Anomalocaris, that aren't totally direct though, and that's because the brain shape of Jianfengia actually connects it to mandibulata. That's things like crustaceans and myriapods, meaning centipedes and millipedes. So yeah, there's a lot of brain stuff, and again, I'm not really well versed on arthropod brains, but I hope to be soon potentially. But really interesting that we're able to start understanding a lot of the evolution of the arthropods just based on brain shapes. And then jumping into chemicals and how fossils are preserved, different elements will respond differently to light. Basically, the electrons will jump up in energy level and then back down, and when they go back down, they can release a little packet of light called a photon at a specific wavelength. And those can vary depending on the light coming in. This is where micro X-ray diffraction comes in. You basically shoot the fossil with X-rays, and the elements in it bounce different kinds of light back. In Triassic rocks, different marine fossils show different patterns. There is some overlap, but based on the scans, if there's some tiny piece that's been very hard to ID, we might actually be able to show what it is now based on these different elements and the methodology produced in this paper. Mazon Creek is famous for Tolly monstrum, but there's other things, including loose associations of material described here as some sort of bromatolite. Basically, whatever animal this was threw up its food, and now we have basically what its lunch was. And this animal had eaten horseshoe crabs. At this time, there were many freshwater members of the horseshoe crab family, so it very well could have been from one of those freshwater animals, but this deposit is kind of brackish, meaning essentially it's a stream entering the ocean, so it could very well have influence from both. What they've also suggested is potentially it was a lungfish or something related. When you think about lungfish, oftentimes we find just their tooth plates, and these are really good for crushing things. Potentially even things like horseshoe crabs. Also, here's one tooth from the Chinle Formation. Next one is jumping way up towards modern times in the Grave Fossil Site in Tennessee. So, hi Derek. Hi Will. This isn't about their work, but rather deer, and the deer at the Grave Fossil Site is odd. It's similar to Eocolius, though a bit different in a few traits, which can be seen in this graph. However, the broad similarities do also mean it is still Eocolius, just a different population accounting for those differences. Other early North American deer come from Florida and Washington and are just slightly younger than the Grave Fossil Site deer, meaning that they rapidly covered all of North America and were probably being kind of like white-tailed deer and just browsing everywhere on whatever they could find. Electron microscopes can see small things really, really well, and fossil pollen is really, really small. Angiosperms are fruiting plants which show up in the middle to late Jurassic and then diversified in the Cretaceous. However, a lot of this diversity doesn't really seem to be from modern groups. Looking at pollen can help to show this, where only during the very late Cretaceous does pollen resembling modern groups really start to appear in large numbers. And then again, after the KPG extinction, a lot of that diversity that wasn't modern trees died out, and then those that survived became the modern trees we do have. But at least for a long period of the early angiosperms, there wasn't anything resembling modern forests, despite angiosperms still being present. And this is finite element analysis on ring reptiles, which is basically what you expect. The pointy ones face more pressure at the tips, whereas things like globidens are more distributed. It's what we expected, but now we have actual data. But also there's things that we didn't expect, because they didn't just do normal FEA using that model, but also did a mesh-weighted arithmetic mean, which means basically you make a mesh of the different faces on the tooth and then add up the different stresses across those entire areas. So it's a new method of FEA that's been used to try and analyze how these teeth might have performed. And in some cases they did perform slightly differently than what was expected, but that may also just be the model needing to be tested more thoroughly. And then jumping real quick to the Permian, and especially in Africa around Zambia, because there was a whole special volume on that. Lystrosaurs is the most famous survivor of the Permian Triassic mass extinction that killed 90% of life in the oceans and 75% of life on land. This paper described 3 new Lystrosaurids, meaning they all belong to the same family that Lystrosaurus does. All 3 are new genera. There's Lystrosaurus balfe, Madum ambisa opanian, and Eutrichognathus kingae. Madum abisa is older than the others and comes from the Madum abisa mudstone formation, and it's pretty common there, meaning it's not just the conditions at the boundary of that extinction alone that made Lystrosaurids common. This is also true because all of these have pretty similar body shapes to Lystrosaurus as far as their head shape, and also somewhere suggest they were feeding in a similar way, rooting through the ground. My wife also often says that these animals are very ugly, but despite that, it's not the extinction that made them that way. They were very well adapted to the conditions that would just eventually become more common during that extinction, and that prepared them for success just through sheer chance. And at least Madhu Mambisa may have had to watch out for Arctops umulunshi, which is a new gorgonopsid from the Madhu Mambisa mudstone formation. This helps to show some connectedness between the Zambian Permian, where these animals came from, and the Karoo Basin in South Africa, which does contain a different Arctops species. Aulacephalodon kapoliwacela is a new geikiid dicynodont. This is a group of dicynodonts which, unlike Lystrosaurs, went extinct at the end of the Permian, and like Arctops umulunshi, it is an already named genus, which has also been found in the Karoo Basin, and hey, there's already a lot of related animals between these two areas. Hopefully there can be some more work done in the Triassic, so you can try and see how these two different populations of animals, those in Zambia and those in South Africa, may have responded differently to the Permian-Triassic extinction. Dicynodontoides kubwa is a new emydopoid from Tanzania, specifically the Usili Formation, which is interesting because it, unlike the material from Zambia, has more like that from Eurasia and not the very well-studied areas in southern Africa. So this isn't a perfect pattern across all different regions of Africa. There is some regional influence coming from different areas, and this is just one example of that, with this animal being more closely related to those coming from Europe. And back to the Zambian Madhu Mambisa mudstone formation, the Captorhinids are a group of tetrapods which are not mammals or reptiles or amphibians. They're kind of probably closer to mammals or reptiles, but it's hard to know for sure. Regardless, emydopoid is one of the last, and it has traits that place it as a morodosaurine. And the way that these have been studied in the past has been heavily biased by characters which change as the animal aged, and this fossil has helped to show that. However, all of the known ones also come from before the end of the PT interval, meaning they were probably already extinct before the Permian-Triassic extinction happened in earnest. Still, very interesting animals that really helped to shape a lot of early ecosystems on land. Cyanosaurus is another gorgonopsid known from across Pangaea, but this is the first from Zambia, and it would have lived alongside a few other gorgonopsids. This one isn't complete enough to be named, it's mostly just a skull, but what this does mean is that with many of these animals living alongside one another, there was some level of hidden partitioning among these predators that we can't really detect exactly how they were doing it, but that they definitely were, because otherwise they'd be competing directly with one another. And seemingly they lived alongside each other without too much issue. And feeling worn out by the Madhu Mambisa mudstone formation, there was a big look at all of the vertebrates across the entire formation. Basically, they went layer by layer and assessed which fossils were found in each. And they found there's at least 2 vertebrate assemblages and 4 depositional environments. This is actually pretty similar to what's been seen in the very well-studied Karoo Basin between their different assemblage zones. However, with the new species found in Zambia, it does mean there's the potential to do a lot of new studies of how these ecosystems changed and may have been different from what was happening in the Karoo Basin in the build-up to the Permian-Triassic extinction. And those faunas would include temnospondyls, which are the group that would eventually lead to modern amphibians. However, back in the Permian, they were much larger, at least in some cases. This group also experienced massive amounts of extinction during the Permian-Triassic, and there is a new one also coming from the Madhu Mambisa mudstone formation. Rhinceps caribensis is related to other temnospondyls from Malawi and South Africa. So again, it's a very regional area, but this group didn't survive the Permian-Triassic extinction despite other temnospondyls in the region successfully doing so. So there is a lot of interest as to why these animals died out while other ones that were pretty similar survived. Gharials don't live in South America, and in fact they only live in a few rivers in southern Asia now, and are critically endangered. However, the fossil record shows that they lived in much broader areas than they do today, including South America. There's even some known from the Pisco Formation of Peru, which is known for having great preservation, and that's the case here, with complete skulls having been found. By placing all of these skulls into the phylogenetic matrix of other fossil gharials and the modern ones, it was found that the modern gharials split from their South American relatives probably around 30 million years ago. During the Ice Age, sea levels were lower, and that means there was a lot of possibility for a lot of migration, except for in kind of the Southeast Asian archipelago, and that's because of things like the Wallace Line, named for Alfred Wallace, who recognized the differences between the Australian continent and Southeast Asian faunas, and was able to show hey, look, there's probably something here that just prevented these animals from migrating across. And that's the Wallace Line, a deep sea trench that essentially means that even though the sea levels were lower, animals couldn't migrate across it. A new fossil from Luzon in the Philippines, though, does suggest that at least some animals, in this case Stegomastodon, an elephant, could successfully cross this Wallace Line. And that's just by island hopping. And frankly, just elephants are really good in the water, surprisingly good, so it makes sense that they were actually able to do this. And they actually did make it pretty close to Australia, which means had environmental conditions not warmed and potentially caused the extinction of Stegomastodon in the area, there potentially could have been an elephant species in Australia. But again, human migrations and climate seemingly just made that an impossibility. The first deer in South America got there about 1.8 million years ago, and now most of them are fairly small, like the brockets and pudu. The La Venta locality in Colombia, though, is older than this, coming from the Miocene, at least 10 million years ago. And now there's a new deer from there, and it's not actually really named. It is just a jawbone, so, maybe it's slightly more robust than modern living deer, but only just. However, based on its absolute size, it could be one of a few fossil and living deer. Regardless, this is good evidence that at least a very small population of deer made it to South America by the early onset of the ice ages, which begs the question, why is there an 8 to 9 million year gap? We don't necessarily know yet, but it is interesting to think about. Punctuated equilibrium is one of the ways we analyze and understand evolution. It's a model of evolution, in contrast to gradualism. Gradualism states that evolution slowly occurs in all of these groups with just tons and tons of little changes adding up over millions of years to change a species. Punctuated equilibrium, by contrast, suggests that stasis is normal. Basically, sure, there's some oscillation, but generally there's a mean for a species that it just hangs around. Then a disruption in the habitat occurs, and then rapid change can occur. This paper looked at punctuated equilibrium in the context of cladogenesis and anagenesis. This is essentially branching when you have different relations between different groups, or if it is just a single straight line if you're looking at the tree. So for example, something like this kind of snail evolved directly into this kind of snail, directly into this kind of snail. Or one little bit of this kind of snail broke off into two separate populations, and one of those eventually died out, and then the other occurred. And both do probably occur to different degrees. The paper presents a number of different kinds of examples, and I'm going to narrow it down to just two. This first one is kind of the anagenesis model, where it's very slow growth of one species into another one, and that's basically it. That is potentially still very rapid change in that time for punctuated equilibrium. However, again, that is an anagenetic change. And then you have ones that are basically just motifs of this one, where you have the single lineage at the beginning, a very rapid change into a secondary lineage, which then those two lineages coexist for a while before the older one dies out. And that seems to be the more regular pattern for speciation found in this paper. Essentially, one population becomes a separate species and eventually overtakes the parent species. By looking at broad examples in the marine fossil record, these researchers suggest that punctuated equilibrium is correct, and that it facilitates cladogenesis, again, meaning those kind of branching events, and the later overtake of the ancestral populations. Rhizoliths are root trace fossils. Basically, the roots of plants will get rid of some minerals, which causes variation in how the sediment consolidates in its appearance. This preserves long tubes of different matrix in the rocks where the root once was. This paper actually looked at them in a systematic kind of way to try and categorize what they can show about the environments that these were found in, and found that there is a difference in upland versus lowland parts of river system floodplains. In the upland part of the river, deposition would happen, there's a bit of time before new life grows, but then small, herby, woody plants would develop. Unfortunately for these smaller plants, this would be unstable due to the occurrences of other flood events. Meanwhile, in lowland flood environments, there's a larger flood plain, meaning there's areas where the stream would override the banks, and then they would rapidly get low-laying plants, and then followed by larger trees colonizing the new substrate brought in by the flooding. This would then be more resistant to secondary flooding because of the larger plants that are able to actually colonize large parts of this new sediment. This is one of the first times that rhizoliths have been able to use something other than, hey look, trees and other plants were here, and shows that their size can be very informative about different depositional environments. Biochemical studies in paleontology can also be useful, like in studies finding the leftovers of cholesterol in Dicynsonia, or the chemical makeup of Tolimonstrum leaking it to chordates and vertebrates. But often plastics are used to consolidate and hold fossils together. These are things like paraloid B72, which dissolves in acetone, can be applied to the fossil, and then as the acetone evaporates, there's just a small layer of plastic left. It's really useful. This paper looked at the hydrocarbon chains in B72 and tried to see if those could affect the chemical analyses, looking for other hydrocarbons in fossils. And in general, not really, no, actually, it's pretty consistent. However, if there is a poor grade paraloid used, that can introduce contaminants. I mean, you shouldn't just be grabbing, a cheap bottle off the shelf of acetone that's not really well produced or of poor grade paraloid B72. You want to order something properly from proper, producers, manufacturers of these chemicals. This paper is pretty straightforward in that if you do more math on the shapes of fossil shark teeth, you can tell more about them. Crazy, I know. I don't know what else to say, as this is pretty straightforward, but this paper used geometric morphometrics, which is points and their relationships to one another as opposed to measurements of different features. Basically just the left versus the right here in this image. And this actually helps to streamline a whole lot as far as being able to actually tell different things about where in the mouth the teeth might have gone or what kind of species the tooth might be from. Styracosternans are a whole group of ornithopods, which would lead to the hadrosaurs and the iguanodontians, in addition to others. This group is known from the late Cretaceous of the Iberian Peninsula, and a handful of fragmentary fossils from the early Cretaceous of Spain have been found to belong to the group. So the story here may not be as simple as they evolved elsewhere and then eventually migrated into Iberia during the late Cretaceous. Essentially, this handful of fossils helps to show that they were there for a good long time. Flatfish are weird. Like, here, you can look at this diagram of how they go from pretty normal looking larvae, very fish-like, to fully rotating their eyes to be on the same side of their head. This is a fossil of Arnoglossus sauvagii, and the genus Arnoglossus is actually still around, living mostly in the eastern Atlantic and Indian Oceans, and this one comes from the Mediterranean. This paper was basically looking to see if Arnoglossus sauvagii was actually a species of Arnoglossus because it was named in 1878 and fish are sometimes understudied. What it found is that, yes it is, but also this is from the late Miocene, about 5 to 8 million years ago. And combined with other fossils of other flatfish, it does suggest that the climate conditions of the Mediterranean at that time were very, very good for flatfish because they actually had higher diversity then than they do now. Crocodilus is the genus for most crocodiles, and Crocodilus sudani is a new species from Sudan and is more related to other fossil crocs than to modern ones. This is interesting because all of those lineages of those African fossil crocs are extinct and just the modern one exists. So basically there was an entirely separate group of crocodiles in Africa as little as 40,000 years ago, and they've just totally died out just by living so recently. Unanlogines are dromaeosaurs from South America, dromaeosaurs being the kind of raptor dinosaurs. This paper, though, found something different. They're not dromaeosaurs, instead they are within aves, meaning that rather than being dromaeosaurs, they are birds. Not even just like, stem birds on the lineage to birds, they're just birds. And that's combined with the Halscaraptorines, these smaller dromaeosaurs that are also very well known for potentially being somewhat aquatic, and that's hard to say, there's a lot of debate about it. This has been found by this team before, so we know it's a result that can be found. However, what's really interesting is there's going to be a lot of debate about this. And that's because some researchers might weigh different characters differently. Oh, you're saying this is a flange on this bone, and I'm saying instead of that, it's just a smaller projection, so I'm not going to code it the same way. Or potentially just thinking different characters are important. But that's kind of how paleontology works. You're looking at different fossils and interpreting them and what's important about them in different ways. So maybe they are birds, but maybe they're not. We'll have to do more research to know for sure. There were also new fossils of Spicomelus, a Middle Jurassic ankylosaur, initially named from a bit of rib with some massive spikes on it, and even then it was known to be an ankylosaur and probably one of the oldest in the group of heavily armored dinosaurs. These fossils show it was even more bizarre though, because it had a ton of other little odd spikes all across its body. Like it just took spikiness in this group to an entirely different level. Like Godzilla universe levels of spiky. Image of Anguirus here next to Spicomelus, Anguirus being from the Godzilla universe. The new material isn't a complete fossil, but based on what we know of the group, they did make this reconstruction. I say that because some of the tail pieces are just more fragmentary, so the interpretation is fine, but I probably just would have been a little more conservative with more of just kind of a bat tail, like a baseball bat or a cricket bat, as opposed to a more spiky tail. That would have been more in line with what we see in some early groups of other ankylosaurs, like the polacanthines, which didn't have quite as large of spikes on their tails. It's also interesting in that it's from Morocco, and it doesn't really group with other things we know from the Jurassic, particularly of North America, as those still would have been fairly close. Instead, it groups with Chamosaurus, Chonqinglong, and Gobisaurus, all of which are from Asia. Meanwhile, Spicomelis, as I said, is from Morocco, and at least some time after the breakup of Pangaea, although North Africa and Western Eurasia still had large amounts of faunal interchange, allowing for that migration of early ankylosaurs from Africa to other continents. Alternatively, they could have just evolved somewhere else even earlier and shown up everywhere, and this is just the first one we have due to the incomplete nature of the fossil record. Also, there's a number of traits that are shown to have developed at different times, such as a more developed sacral shield, meaning essentially it's just this big shield part over the hips. For an example, you can see this part of Gargoylosaurus from North America. There's also more specialized tail vertebra for making the tail into weaponry, and that's actually lost in nodosaurs, which is why they were more likely fighting with their large shoulder spikes than tail weaponry. Although that's not consistent. In fact, Gobisaurus, related to Spicomelis, lost the tail weapon entirely, but kept the kind of handle vertebra that would have helped facilitate a tail weapon. Moral of the story is ankylosaurs are weird. Really hope we get some more early ones, just because their evolution is very strange. Basically, we very rapidly get almost all of the late Cretaceous groups, and we don't necessarily know how well they actually diversified before that. Hey everyone, thanks for watching. If you really want to support the channel, feel free to go check out our shop at raptorchatter.com. We have all kinds of wonderful things, like the shirts that many people have asked where we've gotten them. Now you can have one too, from us. Yay for supporting the content creators you like. Also the perfect time to look there for holiday gifts for whoever you might know that likes paleontology. Presumably somebody does.